cell performs the important events of the cell cycle in an organized series of steps. In this particular video, we are going to look into these steps. So, let's jump in. In a typical eukaryotic cell cycle is divided into two major stages that are interphase and mitotic phase. Let's begin by examining interphase, which is derived from the prefix inter, meaning between, and phase, meaning stages. Yes, because interphase refers to the period between the end of one mitotic division and the beginning of another, while it was previously referred to as the resting phase. The term was later changed because the cell is actively preparing for the next division by undergoing both cell growth and DNA replication, rather than being completely at rest. Due to this reason, it is also termed as the preparatory phase, and approximately 95% of the cell cycle is spent in interphase. Well, this interphase is not that simple, but it further includes three phases that are G1 phase, S phase, and G2 phase. Well, G1 phase is also known as first gap phase, first growth phase, or post-mitotic gap phase, which is, in fact, the interval, or gap between mitotic phase of the previous cell cycle and the DNA synthesis of the current cycle. G1 is typically the longest and most variable cell cycle phase. Its duration varies, even among the various cells of the same species. However, G1 phase might last about 11 hours. During G1, the cell grows back to its normal size after dividing and makes sure it has everything it needs to start dividing again. Because, you know, a cell won't divide if it doesn't have everything it needs. So, cell increases its supply of proteins, increases the number of organelles, such as mitochondria and ribosomes. But do remember that it does not replicate its DNA. Now, in G1 phase, a cell has three options. One of them is, of course, to continue with the cell cycle and enter the S phase, where DNA synthesis occurs. The other one is to stop cell cycle and enter the resting phase, called G0 phase, where it becomes quiescent and may undergo differentiation or other specialized functions. An example being a mature neuron in the brain that has completed its division will stop the cell cycle and enter the G0 phase. It will remain in this phase for the rest of its life, performing its specialized function. And the third one is to become arrested in G1 phase, which can occur if there are problems with DNA or if the cell receives signals indicating that it should not proceed with the cell cycle. In this case, the cell may enter the G0 phase or may eventually re-enter the cell cycle if the issues are resolved. Well, the deciding points is referred to as the restriction point or checkpoint, and it's like a control system that makes sure the cell is ready to divide. So that was about G1 phase. Now, the next phase is the S phase, or synthesis phase. Name is giving you a hint that there's some sort of synthesis going on here. Yes, that's because the DNA synthesis takes place in this phase. The cell's chromosomes are replicated. Now, you know that human cell has 23 pairs of chromosomes. That makes a total of 46 chromosomes. Well, till S phase, all these 46 chromosomes have one chromatid each. Now, on completion of the synthesis phase, all of the chromosomes have been replicated. That means each chromosome consists of 2 sister chromatids. But keep in mind that the total number of chromosomes remain the same. That means 46 chromosomes, or 23 pairs of chromosomes. Also, another important thing happening here is that the centrioles in a centrosome undergo replication, resulting in the formation of 2 centrosomes, each containing a pair of centrioles, which play an essential role in the organization of microtubules during cell division. Rates of RNA transcription and protein synthesis are very low during this phase. An exception to this is histone production, most of which occurs during the S phase, to package the newly replicated DNA into chromatin. Now, this S phase usually lasts about 8 hours. That concludes our discussion on the S phase. Let's now shift our focus to the next stage of the cell cycle, which is the G2 phase. Well, the G2 phase is also referred to the second gap phase, second growth phase, or pre-mitotic phase. It is the interval, or a gap, between the completion of DNA synthesis, or S phase, and the cell division, or mitotic phase. And in a rapidly proliferating cell in humans, it takes about 4 hours to complete. You can see that it's a brief period, and during this phase, cell prepares to go into the mitotic phase. So, it grows more and makes RNA, proteins, and other macromolecules required for multiplication of cell organelles, spindle formation, and cell growth. Now, when these activities reach a critical threshold level, the cell enters mitosis. Along the way, the chromatin and cytoskeleton are prepared for the dramatic structural changes that will occur during mitosis. If damaged DNA is detected during G2, a checkpoint activates the DNA damage response and delays entry of the cell into mitosis. So, that was about the interphase. That includes G1, S, and G2 phase. Now, moving on towards the M phase, or what we call mitotic phase. Well, the M phase is about 1 hour in a rapid proliferating cell in humans. During the mitotic phase, the cell separates its DNA into two sets and divides its cytoplasm, forming two new cells. This phase basically involves two processes. One of them is the nuclear division, which is also called as the karyokinesis, during which the nucleus divides to form two daughter nuclei. This includes the process of mitosis. And this nuclear division is then followed by the division of cytoplasm, which is referred to as cytokinesis. So, mitosis and cytokinesis together define the division of the mother cell into two daughter cells, genetically identical to each other and to their parent cell. The process of mitosis is divided into different phases, which are known as prophase, metaphase, anaphase, and telophase. But hold on, I'm not going to bore you with the nitty-gritty technical details of mitosis, at least not yet. If you really want to dive deep, we've got a fantastic video on mitosis over on sqadia.com. But first things first, can you remember all the phases of the cell cycle in order? If you're struggling to recall, fear not, my friends. I've got a foolproof mnemonic that will make it easy as pie. It goes like this. Great scientists get prizes more akin than chemists, where each letter represents a different phase of the cycle. Like G is for G1 phase, S is for synthesis phase, and G is for G2 phase. These three completing the interphase, and P refers to the prophase, M is for metaphase, A stands for anaphase, T indicates telophase, and C is for cytokinesis. These five indicate the mitotic phase. Once you've got these phases down pat, you'll have a solid foundation for understanding the ins and outs of cell division. So, here, we are done with the cell cycle. Now that you've got the hang of the cell cycle, let's delve deeper. Find out more about the checkpoints of the cell cycle. Stay tuned for the next video in this series to learn about these.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.